As always, WESTalks is a collaboration between IC Impacts and UBC Future Waters, but I don't need to go through that anymore. It's been 50 weeks or so you've been hearing that. The organizing committee, as always, Abhishek, Lili, Fuhar, Jaskaran, myself, and Furia is helping from IC Impacts. Once again, here's the check-in map for WESTalks from all the people from around the world who've listened into our presentations. If you haven't done so, I'll post the link in the chat box, but you can click on there and just check in. There's a red plus button on the top right. All right, so here we are. Dr. Leland Jackson from the University of Calgary is our speaker for today. We have one more next week, Dr. Amit Pinto. And then the West Conference is the week after that. And all previous WESTalks are available on the IC Impacts YouTube channel. So just go on YouTube and search WESTalks and you'll find all of those. Okay, so today's speaker is Dr. Leland Jackson. So Dr. Lee Jackson is a professor of aquatic ecosystems and communities in the Department of Biological Sciences at the University of Calgary, and the scientific director of Advancing Canadian Wastewater Assets. Research in his lab centers on understanding relationships between structure and the function in southern Alberta lakes and rivers, and particularly internal, so the food web, or external, climate, drivers that lead to rapid change or confer stability. Current projects in his group involve the creation of a data collaboration platform for the Bow River Basin, along with IBM, determining microplastics' fate in municipal wastewater treatment, genomic assessment of microplastic-associated biofilms and wastewater treatment plant microbiomes, assessment of NIF genes, so nitrogen fixation, across salinity gradients, contaminant effects on and development of the diagnostic biomarkers for native fish populations, exchange of antimicrobial resistance elements during biological nutrient removal, and the structuring role of macrophytes in shallow lakes and rivers. So quite a lot of work there. And with that, I will hand it over to Dr. Jackson, who hopefully screen sharing is all good there.
Yes, let me just go find it.
Somewhere in there.
Hopefully it's up now.
Yep, we can see it in, yep.
Perfect.
Mode. There we go. Yeah.
Perfect. I'll just switch over to the full screen. Great. Well, thanks for inviting me to talk today, and thanks for that introduction. I did take a look at some of the past talks, and it looks like you've got a really nice variety of people talking on a whole range of subjects. So hopefully this fits in. I suspect it might be a little bit different than what you've heard in the past, but that's okay. It's good to sort of think broadly, I think. So what I'll do, I was told, 20, 30 minutes. It'll probably be closer to 30 minutes. I'm going to start by kind of outlining a conceptual framework for alternate states or regime shifts in aquatic ecosystems. I'll talk a little bit more about what that means in a moment. I'll give you some evidence from shallow lakes in Alberta and the Bow River, which can display the same kind of features. I'll describe a little bit about advancing Canadian wastewater assets, and then talk about what I see as some of the emerging challenges facing ecosystem and environmental managers, and then what some of the solutions might be. And so I think everybody is probably aware that a vexing issue for environmental and ecosystem managers is trying to understand how ecosystems today are going to respond to environmental change. And of course, climate change is the big one that we're a bit focused on, but there are other sorts of changes that happen too. There are land use changes that lead to changes in nutrient inputs. And so it's not just climate change, but certainly that's one that's in everybody's mind. And the issue is not so much that there can be change in aquatic systems as a function of changing climate or something else, but sometimes there are some really, really big surprises. You get rapid, large-scale change in response for a very small-scale change in an environmental driver. And that's a nightmare if you have to manage lakes or ecosystems, because it's very difficult to see those coming, and that's where the problem lies. So let me start with the conceptual framework then. And it's really common to refer to it as a ball and cup model. And so on the bottom here, I've just put time. And if you look at the cup on the left with the green P in it, this can be the average chlorophyll concentration in a particular lake over some number of years. That's the ecosystem state, and I've just called it water greenness for now. And we know that the system can sort of rock back and forth in this local basin of attraction, or in the case of chlorophyll, it would be some average value plus or minus a measure of variability. And then for some reason, the state can flip to a different situation or a different configuration. And so if it's greenness, we go from sort of a low greenness state here to a high greenness state here. And the system can stay in this high green state or this high turbid state for a long period of time. And so people are very, very interested in trying to understand a couple of things. What causes the state to flip from this state up to this state? What are the drivers, and how big do those drivers need to be? And when you're in one state or another, what are the elements that allow the state to remain there, so that it's not simply a matter of just pushing the P back over this little peak and it'll drop all the way down to here. So what are the stability elements that, or the things that confer stability to that state? And that's where the stable part of alternate stable states comes in. And shallow lakes are great systems to work on this because they're diverse. We understand a little bit about how they work. And if you have enough money, you can actually manipulate them and test hypotheses about what those drivers are. And I'll talk more about some of those in a moment. And so in the shallow lake literature, which is a fairly mature now, we refer to a clear state and a turbid state. And those, of course, are relative terms, but I'm going to draw on those a little bit more. So here is a sort of the ball and cup model, but it's rearranged a little bit differently. So here's a turbid state. Here's turbidity in nephelometric turbidity units. This is just a measure of water cloudiness, and it includes everything that scatters and absorbs light. And here's a gradient of nutrients along the x-axis. And so over time, lakes start from their formation at very low nutrient status, very clear. And naturally over time, as the watershed weathers, they move along this axis and they tend to become greener. We, of course, accelerate, we meaning people accelerate that in some cases, but if you're in the mountains, those lakes are undergoing that natural process. So here we have a turbid state. This is the high turbidity, the cloudy state. Here we have a clear state, the low turbidity or relatively clear state. And this critical turbidity is really the distance between here and here. This is what we call in mathematical terms the separatrix, but this is the sort of gradient that has to become overcome to flip a system from this state up to this state. And you'll see I've added a couple things to this reorganization of that ball and cup model. One, this is without macrophytes. So these are the freshwater seaweeds that grow in lakes. And this is with macrophytes. And so I'll show you a little bit more about this in a moment here. But the arrows indicate that when a system is within one of these states, there are stabilizing features that in this case, the turbidity moves off the long-term trend, stabilizing features tend to push the turbidity back to that long-term trend. And a really important thing to note here is that if we start at low turbidity or low greenness over time, and gradually with an incremental increase in nutrients start to creep up towards this critical turbidity, you can have a very, very small change in nutrients right here that will all of a sudden flip the system up to this turbid state. And that's where the big surprises come if you happen to be a lake manager. It's this really, what seems like a very small change in nutrients, but all of a sudden you have a massive change in the configuration or the state of the response system. So this is all well and nice in theory. The question is, does it exist in real life? And population and ecosystem ecologists have done all sorts of modeling over time to show that in theory, these regime shifts or alternate stable states are possible. But the evidence for them until I would say the last 15 or 20 years hasn't been present in abundance. So here is evidence for a couple of different regimes in shallow Alberta lakes. So there's 28 lakes in this plot. And the first thing to notice is this is maximum depth of the lake. So I've divided the systems here at about 6 meters maximum depth, because it turns out if a lake is 6 meters or deeper, it will thermally stratify in the summer. If it's around 4 meters or less, it will continue to mix all summer long. So that's polymictic versus stratified. And the turbidity is here on the y-axis. And the way to read the code here is the size of the circle is the proportion of lake bottom that is covered by macrophytes. And it's in 20% bins. So if we just look at the deep lakes, you can see that all of them have very small circles, meaning 0 to 20, or at most 20 to 40% of the lake bottom supports macrophyte growth. And the depth that macrophytes grow at is often limited by light penetration. So once you get below a certain depth, there's not enough light to support plant growth. So light can penetrate to the bottom of shallow lakes if they're 4 meters. And hence you see a large proportion of lake bottom supporting macrophyte growth. The color of the fill is proportional to the biomass of the macrophyte. So if you're at very high biomass, it's a black fill. If it's very low biomass, it's a lighter fill. And then there's some intermediate ones that don't show up as well on this. These dark fills are up to 5 kilograms fresh weight per meter squared. And for context, if you're swimming through that, you can swim through it. It's going to be difficult. If you're wearing scuba gear, trying to swim through it, you're going to have a really, really tough time at 5 kilos per meter squared. So we're not going to worry about these too much, because these are sort of the traditional fishbowl lakes, but it's really these ones that are interesting. And what this shows is that there is a split in turbidity between high turbid lakes here and low turbid lakes here. Roughly the same proportion of lake bottom is covered by macrophytes by and large. But you see that down here in the clear lakes, there's a lot more dark fill than there is up here, meaning there's a lot more macrophyte biomass. And so the question is, well, do we just have a couple of coincidental clusters, or is there really something here? And to statistically examine that, I use Dixon's test for outliers. And so what you do is you take the highest turbidity value, you subtract the second highest from that, and you create an interlake turbidity difference. And then you go to the next pair and you do the same thing, and you go to the next pair and you do the same thing. And you come down here and you calculate a pair between these two lakes, and you end up with a plot. It's n-1 of the ranked order of interlake difference in turbidity. And here you can see that split between those two clusters. And then what Dixon's test does is it uses this distribution of observations, and it asks, is any of these individual observations an outlier given this distribution of observations? So it's using the actual data, not comparing the distribution to some assumed normal distribution or something else. And it turns out there is one significant departure, and it's this event right here, ranked order number 9. And no surprise, it's between these two lakes right here. And so this is statistical evidence for bimodality in the data set. And that's really the key feature in distinguishing regime shifts or alternate states. Do we have evidence for bimodality? And over time, these lakes have moved a little bit in terms of their chlorophyll value, but the clusters have remained pretty consistent. I'm only presenting 1 year of data here. And so if we look at some functional relationships, there's a couple of interesting things. Here's dissolved phosphorus. Early in the year, the greenness of the lake, and that's represented by the chlorophyll A value here, is determined largely by the amount of available phosphorus. And you can see that when we classify lakes as turbid or clear, the closed squares are turbid and the open squares are clear. We have two very similar relationships, with exception of the turbid lakes are greener for the same amount of dissolved phosphorus. So this is an analysis of covariance that shows that the slopes of the lines don't differ from one another, but the intercepts do. And so there is a higher yield of chlorophyll per unit of phosphorus in turbid lakes, the black squares, versus clear lakes, the open squares. And so you might think, well, that's what's driving the difference in water clarity in these sets of lakes. But if we move down here and now look at the turbidity, which is the chlorophyll plus anything else that's in the water column, like suspended sediments, we see something very different. Here across a range of chlorophyll from 0 to about 10 micrograms per liter, we have the clear lakes. They have very low turbidity across this range of chlorophyll, and the turbid lakes have very high turbidity across the same range of chlorophyll. So there's something besides chlorophyll that's generating turbidity, at least across this range. We don't know what would happen in the clear lakes if we could extend this line out here, because we don't have lakes out here in this area. And it turns out it's suspended sediments that are making the difference there. And those macrophytes are important because what they do is they create physical structure in the water column that does two things. It provides refuge to large-bodied zooplankton that are the very effective grazers on chlorophyll, protection against their predators. So it could be large invertebrates or minnows. And we know that when the macrophytes are present and they reach a certain biomass, they're very effective at trapping particles and preventing their resuspension. So they have a positive feedback that as the macrophytes get thicker and thicker, they trap more particles, they prevent resuspension. The water remains clearer, the macrophytes grow better because there's more light, and it creates that internal structuring feature that you saw in the little arrows a couple of diagrams ago. And so we see something really analogous in terms of rivers. This is data from the Bow River and an analysis that shows now two regimes or two states. I've called them a low quality state on the left-hand series of arrows, and a high quality state on the right-hand series. Upstream, we have very low nutrients. This is Calgary, the footprint of Calgary. Upstream, we have very low nutrients, low macrophyte biomass, partly due to low nutrients. Potentially the substrate isn't very hospitable to macrophytes, and very low turbidity in the water column. So very clear water. And then downstream, we have the opposite of that. We have higher turbidity. We have much, much higher macrophyte biomass. And so if we sort of think through the argument here, if we look at the low quality state, which is representative of downstream, we also have 3 wastewater treatment plants discharging nitrogen and phosphorus into the river. So the population discharges nutrients, as I think everybody knows from wastewater treatment plants. This fuels macrophyte growth. The macrophyte beds trap suspended sediments and reduce water flow. There's an internal feedback that happens. But one of the consequences is that during the night, there's reduced oxygen concentration due to respiration in those weed beds. So as the macrophyte biomass increases over time, this is time on the x-axis, the macrophyte biomass goes up, and we see low dissolved oxygen changes at low macrophyte biomass. But as those weed beds build up, we start to see very large amplitude changes in dissolved oxygen swings. The reason that's important is that young fish that are using those weed beds as refuge need to either succumb to low dissolved oxygen, or they need to get out of those weed beds and move out into the edges and open channels. And when they do that, they're susceptible to predation by larger fish. So increased predation, higher mortality, and at really high DO swings, you can actually initiate summer fish kills within the river. Upstream, the macrophyte biomass has remained low because the nutrient drivers aren't there to fuel its growth and have that positive feedback. And so over time, we see no evidence of large amplitude DO swings. Refuge for young fish is in rocks instead of weeds. We don't see mortality. We don't see summer fish kills. So this is a high quality state. This is a low quality state, and that's from a fish perspective, right? And so when we put terms on quality, of course, it depends on our perspective. Anglers would certainly agree with this, but other people may not necessarily agree with this characterization of high and low quality. And so we see very analogous things happening in the Bow River and other rivers in southern Alberta that we see happening in shallow lakes. And so there's a lot of similarities, not only in the conceptual framework, but even some of the mechanisms that are at play. And it turns out that the aquatic macrophytes are a very key component to that. So that's sort of the conceptual framework. That's evidence. A challenge if you're a manager is trying to understand where you are in that. And if you are managing the Bow River and you're managing nutrient inputs, You can't control the weather, you can't control year-to-year flooding and changes in nutrients that are due to suspended sediments. How do you know when you're getting close to potentially a massive summer fish kill that has tens of thousands of fish on the beach versus not that? That would be the worst nightmare for Alberta environment managers who are charged with managing and setting limits on nutrient discharge from wastewater treatment plants. So here's kind of my view of wastewater utilities. The mantra is sort of everybody's dying to be second, right? So nobody wants to allow scientists and engineers to go into wastewater treatment plants and play with processes because they don't want to upset those processes, but it's okay to go do it in somebody else's wastewater treatment plant. And so this leads to a real problem. If nobody's willing to be first, how can we play around with treatment processes and decide what level of change is necessary to protect its receiving environments? Well, it turns out we built Aqua to address that very issue. It's infrastructure embedded within infrastructure that's designed to be pushed, pulled, and broken. And the infrastructure is in two places. At the U of C campus, we have a microbiology laboratory. All the sequencing is done there for genetics analyses. We have an aquatic laboratory where we look at dose-response relationships and combinations of chemicals to develop diagnostic tools of exposure and understand how they work. And we have an isotope laboratory, stable isotope laboratory to do tracing studies. And then down at Pine Creek, we have a wastewater treatment plant that's embedded within the City of Calgary's Pine Creek wastewater treatment plant. We did that so that we could test advanced technologies, push them to failure. Of course, engineers don't say push them to failure, they say test the design limits, but that's what we do. We push them until we break them to understand where things are when they break and what happens. And we have 12 naturalized replicate research streams that we constructed. And we have an analytical laboratory that has some of the best and most comprehensive analytical capacity in Western Canada and possibly even North America. So here's physically what the site looks like. In this analytical laboratory, we have an alphabet soup of analytical equipment. If you're interested, you can go to the Aqua website under the laboratory and see the various equipment, but it's mass specs to measure volatile, semi-volatile, non-volatile species of chemicals and trace metals. We have our experimental wastewater treatment plant in here, where we have advanced oxidation and membrane-based modules that we can look at contaminant removal with the analytical chemistry, what goes in, what comes out, what works in tandem. We can look at disinfection byproducts. We can look at membrane fouling and understand that. And then we can send water from two of the modules here to the research stream. So we run Bow River water as our reference condition. Water comes from an intake building down here, is pumped to a head pond, and then from that head pond, it's distributed through here. So this is what the streams look like at construction, commissioning, and this is what they look like today. We've allowed them to colonize naturally. They've been invaded by willow, sandbar willow, cattails, sweet clover, poplars. There's abundant growth within the streams, and even some fish have snuck in through the screens in our intake structure. Screens that were defined by DFO, we've had small fish that have snuck through there and made their way up to the head pond and set up shop. And they've even gone from the head pond through all the piping and are now living in the stream. So we know that because we shock the streams pretty much annually to try to remove the fish that we don't want there, so that the fish we do want there, which are planted, either put in and allowed to free range or put in cages more commonly to look at exposure effects. We know there are other fish in there besides that. So I'll just run through the plumbing really quickly because I think it's important to understand how we set this up. And then I'm going to come to this whole notion of if we are going to be first and do experimentation, how can we use science to inform changes in policy and regulations? And what are the things we're trying to influence or participate in? So water comes in from that intake. It goes through a screen and drops into a wet well, and then it's pumped up to the head pond, which is 5.5 million liters. Fish and macrophytes that get drawn in through the intake structure go around in a loop and go back into the river. We take water from secondary clarifiers, so it's biological nutrient removal at Pine Creek. We take the clarified water, it goes into our wastewater treatment plant. It's first processed through an ultrafiltration module. 0.02 microns is the membrane size. And then from there to one of three other modules. So an RO system or nanofiltration, 0.001 microns or 10 nanometer pore size, an ozone module or a peroxide module. And both the ozone module and the peroxide module can be run with or without UV radiation to essentially supercharge the formation of the hydroxyl radicals that are doing the real work here. So membrane-based systems, advanced oxidation systems, and I lump both of these into AOP systems or AOX systems. I don't distinguish. Then water from two of these modules can go out to the streams, process A or B, and can actually dose the streams to look at the whole effluent effects of taking treated water, whether it's from two of these modules, Pine Creek final effluent, which is today's gold standard, or the reference condition, which is downstream from about 430 million liters of effluent discharge into the Bow River. We can look at all of those in a true controlled experiment fashion. The foundation of modern biology is the controlled experiment. And so we can set that up by doing dosing and then running these streams in triplicate. So water from the head pond goes through a distribution gallery, gets the effluent added to it, and then runs through the streams. Depending on the nature of that water, if it's permitted for discharge, so final effluent, or Bow River water is back into the river. If it's from our treatment plant, it has to go into the headworks of the Pine Creek facility, be treated, and then released. And we've set up the streams in this manner, 320 meters long each, with 10 pool riffle combinations, so we can look at the fate and effect of chemicals as they move along the stream, who takes them up, how are they transformed, are there different exposure concentrations as we move along this, gradient, and what are the consequences? And we can also in the distribution gallery add individual compounds if there's an emerging compound of concern that we want to trace. And so when you put it all together, it looks something like this. And the scale here is really at this divided ecosystem. So I'm not a big fan of bottle experiments. They offer high signal, high precision, high replication. But we have 50 years of data from dose response relationships. If you go to a CTAC meeting, you're going to see that. But I can't take any of that information from these bottle experiments and go to the Bow River and figure out what that's going to mean. And that's because there isn't one chemical present in the Bow River. In fact, it's many thousands of chemicals. And there are all sorts of things happening in the natural world that generate noise and variability that have nothing to do with individual chemicals. So we constructed those streams in a way to reduce the two largest sources of environmental variation, stream morphology and flow variation. And we've eliminated those. And in doing that, we have effectively increased the signal to noise ratio so that we can do exposures at environmentally relevant concentrations and develop and assess diagnostic tools. I sort of think of it as, short of a nuclear bomb, what can you actually measure in the real world? And so we've tried to remove a lot of the natural variation and noise to bump up that signal to noise ratio. The other equivalent to whole ecosystems here would be the experimental lakes area in northwestern Ontario, where whole lakes are set aside to do exposures. So here's the distribution gallery. We have complete control over the mixing, and this little ball valve that is kind of sitting in the front of each of these galleries is to set up a carboy, and then with a peristaltic pump, you can just dose an individual compound in here. It would go into the head of the stream, and then you can trace or follow it as it moves through the stream. It could be a new chemotherapy drug, for example, that has particular properties that would cause it to interact with endocrine systems. We might want to understand what the consequences are to environmental exposure. So here's Lee's laundry list of challenges that we're sort of currently dealing with, have been dealing with, and will be dealing with. The largest problem globally in terms of water quality, I think, still involves nitrogen and phosphorus and eutrophication. The greening of waterways and creating water quality that is not suitable for consumption or is poor quality for consumption. Blue-green algal blooms are happening with increasing frequency around the world. They're driven by high phosphorus availability and those NIF genes that were talked about earlier are the nitrogenase genes that allow blue-green algae to take N2 and split it and then do work with it to grow more cells. Blue-green algae, or a number of them, also produce a very nasty suite of toxins called the neurotoxins and hepatotoxins called microcystins. And so that's another aspect of poor water quality that happens besides creating anoxia and a whole host of things. Biosolids management from wastewater treatment plants is gaining more attention. The more we start to delve into it at Aqua, the more we're finding it's full of all kinds of things that we probably would prefer aren't there. And so what you do with these biosolids, and right now, in many places, biosolids are sprayed on fields as a nutrient amendment, but they contain a whole host of metals. The silver that's in our antimicrobial preparations, in my son's hockey gear, and jerseys that keep his hockey bag from getting really stinky, that are in water bladders in my wife's dental office that keep the water system from developing a bacteria growth. All of these things, many of them contain silver. That silver ends up going through the wastewater treatment plant. It turns out it's ending up in biosolids. Microplastics are ending up in biosolids. There are biofilms growing on those microplastics that are hosting all sorts of antimicrobial resistance. So a whole host of challenges associated with the biosolids management. As we find fewer and fewer water supplies, and climate change is changing water availability, and basins are being closed, it's maybe a bit hard to, on the West Coast and in Vancouver today, when you're expecting to get 30 millimeters of rain dumped on you, to think that there could be water shortages. But the South Saskatchewan River Basin has been closed since 2008 to new water withdrawals. So if we were to build an industry somewhere within the SSRB and we needed water to pursue that industry, let's say it's a new meat packaging facility, we would have to go and buy a water license from an existing water license holder. You can't buy that from the provincial government anymore because there are no more to allocate. And so as we go forward and we have more industrial growth and our cities get bigger and we all use water, we're going to figure out how to do more with less. And water reuse is going to become more and more involved in the conversation of how that might happen. We actually did a pilot project with Village Brewery and Xylem Technologies, which is one of the big membrane producers, where we took raw sewage, we treated it, and Alberta Health Services was involved as well. We took raw sewage, we treated it, we got it to better than potable water standards, and we brewed Village Blonde with it. And so we canned 4,000 cans. They were sold out in 10 days. The public has an extreme appetite for this kind of novelty thing, and also for thinking about ways that we can make our industries more sustainable. And so we did a whole bunch of webinars. This project was in the top 5, according to Water Canada, for wastewater projects last year in Canada. And this was the #1 media thing that happened at U of C last year. We had close to $1 million equivalent of media outreach by different things picking up this story and processing it, or sort of putting it on their various accounts. So over 400 different outlets use that. We talk a lot about microplastics, but it turns out what people in the plastics business are really worried about they aren't worried about the microplastics, but it's now nanoplastics. And this creates a whole suite of challenges. We have a hard enough time with microplastics because there is no standard way to look at them and measure them and quantify them. It's even harder to look at something that's 1,000 times smaller in size. And so I'm not sure how we're going to deal with this one, but this is where all the discussion is. On the chemical front, of course, it's PFAS and PFOA. We only have 2 compounds regulated in drinking water, but I can tell you that industries are getting really, really concerned about PFAS, these fluorinated compounds that have been characterized in popular media as forever compounds, because they resist environmental degradation and are present in the environment in really, really for really long times. And we know a lot of them have negative health consequences to people. They're endocrine disruptors, they're carcinogenic, they're mutagenic, they have a whole host of nasty side effects to people. And so the chemical companies like 3M and Dow that have used these in the past are growing increasingly concerned due to the long residence time and the demonstration of adverse environmental income, that just like the tobacco industry and the host of compounds in tobaccos that cause cancer and other problems, there are going to be class action lawsuits down the road regarding PFAS. And of course, you've all heard about antimicrobial resistance. Turns out that superbugs aren't just produced in hospitals. There's extremely strong evidence that the biological nutrient removal process is ground zero for the production of AMR by pathogens that you and I release when we're sick and go to a wastewater treatment plant and interact with all the bacteria that are doing the work we want them to do. And then I know some of you know Majid Moshani quite well. Majid and I and Stephen Liss and a few others have been working on this concept of distributed systems. So it's all well and fine to have centralized treatment systems in Calgary or Vancouver where pipes from all over the place bring waste into a one location or two or three locations to treat. But what is happening to remote and northern communities? A lot of them are completely left by the wayside because they can't afford a $400 million wastewater treatment plant and all the plumbing that goes with it. And so I think this is a big challenge, is creating distributed systems that work at the scale of 500 to 1,000 people or a bit larger, are affordable by those communities, can be run by those communities. And we have a lot of ideas about how we might contribute to this. This isn't a technology challenge at all. We have the technology and we've shown it with this reuse beer product, that we can take pure raw sewage and turn it into extremely, extremely clean water. 23 log units of reduction in the pathogens with the treatment processes we used. So 1 times 10 to the 23 reduction from the front door to what was being put in the beer. And so we have the technology, but we have some social barriers that are really getting in the way. But we think we have some ideas about how to potentially overcome those. And if you've talked to Majid about RezoNet and some of the success stories there, you will have a sense for what some of those success stories are. So what are the solutions if you're an environmental manager? And I'll just finish up with a few things here before answering questions. I think solutions require an understanding of cumulative impacts. A kilogram of phosphorus doesn't do the same amount of work in the Bow River from Bow Glacier to Lake Louise to Banff to Canmore to Calgary to downstream where it joins the Oldman River and becomes the South Saskatchewan River. And so we really need to understand, if we're following a cubic meter of water from the source to the ultimate sink, how does that cubic meter of water change? How does, in this case, I've used phosphorus as an example because it does cause greening upstream, but further downstream where there's high turbidity, you don't get the same yield of chlorophyll for a yield of for unit of phosphorus. We need to think about how to put this into a framework that sort of incorporates the biological, physical, and chemical processes and creates regulations that are responsive to those. We require diagnostic tools and biomarkers that assess complex mixtures and get away from this one chemical at a time approach. This is not going to work. We know this. And to continue to try to manage effluent one chemical at a time is just a fool's work. So here are 2 chromatograms. This one is 1,000 masses that come from one single analysis of water coming off the secondary clarifiers into the Aqua Research Facility by LC Qtopf. So this is a non-target analysis. 8,496 distinct masses in one sample. And that might not be 8,496 individual compounds, but it's pretty darn close. It's probably 8,400 or more. There are going to be some isomers in there. And so here are just 1,000 of them, up to 30 minutes of retention time on the column and then elution. And so, you know, if we just look at this band of peaks right here, I've plotted them down here. They're in the faint red. And what the colored peaks show are the compounds that we can identify because we know the mass and we've done some work to figure out what we're looking at. But there are many, many compounds in here. We don't know what they are. We have a suggestion based on their mass. We can query a mass table, but there are a lot of times where there are multiple compounds that have the same mass. And so we can't distinguish those from each other until we do some further work. So this is a real challenge, and dosing the streams with whole effluents means we're dosing with all 8,496 masses of compounds and not just adding one compound and seeing the effect. We know that these compounds interact with one another. We know that in endocrine systems in our bodies and in model organisms, these compounds interact with one another. Some of them excite receptors, some of them block receptors, some of them are estrogenic, some of them are anti-androgenic. And so unless we're looking at the pathways and measuring the transcripts and metabolites, we're just looking at bulk responses. And so we can't really understand the mechanisms. And then finally, my plea is always for scientists, engineers, and policymakers to collaborate and work together, because we really need to understand what is possible with engineered technologies. Me as an ecologist, engineers need to understand how different engineered technologies work, whether it's on advanced oxidation, removing compounds, but potentially producing disinfection byproducts, or optimizing coatings on membranes if you work on membrane systems to reduce biofouling. And policymakers need to understand the strength of the science and the limitations to the science, and how that science can help them make decisions. So I really think we need to work together and de-silo. And the whole point of the data collaboration platform that was mentioned, that's taken 4 years to develop with IBM, is to de-silo water, is to get everybody in the same sandbox in a safe place, adding their data so we can explore it and discover new things, as opposed to all going off to the corner of our lab and hiding our data and not making it available. So with that, I'll be very happy to take questions. I know that was a hair more than 30 minutes, but hopefully it has motivated some questions.